Hello, and welcome to part 4 of the epithelial tissues lecture in Arcadia University's BI327 Histology course. Part 4, we're going to take a look at glands and secretory cells, so some specialized cells within the epithelial lining. Now, to take a look at this, the basic concept of glands is that they're going to be cells that are specialized for secretion. And whether we're talking about single cells or groups of cells, they're going to be involved with specific types of secretory products. Now, most of the glands we're going to be talking about are going to be groups of cells, and so what we're going to see is that they develop from the epithelia very early on in an individual's life. And they essentially grow as this budding of epithelial cells going down into the connective tissue and forming a cluster of cells, which essentially are an extension of the epithelial lining. But essentially, they're not along the epithelia. They're going to be diving down and embedded within the connective tissue of that region. Now, there are two types of gland cells that we can take a look at. The first are going to be exocrine glands. And the exocrine glands are basically epithelial cells that have developed from that surface. They've grown down into the connective tissue, but they've maintained a connection with that surface. And so they're going to have a duct associated with it. And so if you follow the surface, go down into the duct, you can trace it all the way down to the secretory cells, and then come up the other side and follow it back up the duct and then back up onto that surface again. And so exocrine glands are essentially going to be secretory cells that release their product into a lumen, and then that lumen is going to drain into a duct system and then dump out either into a cavity or onto a body surface. And so examples of exocrine glands are going to be things like digestive enzyme glands and sweat glands. The other type of glands are going to be endocrine glands. And endocrine glands are essentially ductless glands. They're going to develop in the same way. So they're going to develop from epithelial cells. They're going to dive down into the connective tissue or dive down into a region within the body. But over time, they're going to lose that connection. So they're going to you're going to have a cluster of cells, but they're not going to be attached to the surface. They're not going to be associated with any duct system. But they're still going to be releasing a secretory product. And that secretory product usually has to be distributed through the body or delivered to wherever that target location is. So with the endocrine glands, we're going to be looking at hormone-secreting cells and hormone-secreting glands. They're essentially going to release their secretory product into the surrounding tissues, and it's going to diffuse into the bloodstream and then be distributed throughout the body. And we'll talk about that again throughout this course. Now, in terms of modes of secretion, and we're going to focus in on the exocrine glands at this point. They're going to be the more common ones. We'll talk about the endocrine, the hormonal glands, and cells later on within the course. But the modes of secretion with the exocrine gland is basically they're going to be involved with producing their secretory product. They're going to put their secretory product within membrane-bound vesicles, but in general, they're not going to release their product until they receive a signal, until that product is needed for some type of biological function. And so if we're taking a look at digestive enzyme cells, we don't want to just be dumping digestive enzymes into the digestive tract, dumping it into the stomach or dumping it into the small intestine if there's no food to be digested. It's going to be a waste of energy. It's going to be a waste of resources. And so what's going to happen is they're going to produce these secretory products within secretory vesicles, and they're going to store them there. Now, in the merocrine mode of secretion, we're basically going to be looking at exocytosis. And so we're going to have the secretory vesicles moving up, fusing with the cell membrane, and then dumping their secretory product out into the lumen, out into their duct system where it can be delivered. And so the merocrine mode of secretion we're going to see in areas like the eccrine sweat gland, the salivary glands, as well as the pancreas, producing lots of digestive enzymes. Now, a modification of this is going to be the apocrine mode of secretion. Apocrine mode is basically what we see for secretion within cells within the mammary gland. We're still going to see secretory product that's going to be produced. It's going to be stored within secretory granules, membrane-bound granules. It's going to accumulate at the surface of the cell, but when it's triggered to be released, it's not as simple as that exocytosis. It's not as controlled as that process of exocytosis. And so we're going to release the secretory product, but we're also going to lose some cytoplasm. We're going to lose some other things that are there. So we're essentially going to lose a lot more than we're going to lose in the simple process of exocytosis. So we're also going to lose kind of portions of the cell in this kind of rapid release materials from the apical portion of the cells in this apicrine mode of secretion. Then the final mode of secretion for exocrine glands is going to be holocrine mode. And basically within the holocrine mode of secretion, we're going to see these cells are going to go into a differentiation process. They're going to produce lots and lots and lots of secretory product. It's going to become accumulated within the cell, and the cell is actually going to go through a controlled cell death. And so you can see the nuclei begin to break down because it's putting basically all of its resources into the production of its secretory product. And so in the holocrine mode of secretion, basically the entire cell is that secretory product. The things that are being produced plus everything else that's in the cell are going to be dumped out and become that secretory product. And we're going to find this mode of secretion within the sebaceous glands of the skin. And we'll talk about this again later on within the course. Now, there are two primary types of secretory products, and we're generalizing this a little bit, but at least for this point, it's going to give you a basic understanding as we go through the course and look at this in much greater detail. The first secretory product is going to be a serous secretion. The serous secretion is going to be a proteinaceous secretion, and it's going to be a more watery secretion. In contrast, the second type of secretory product is going to be a mucus. A mucus is sulfated and silated mucins, so it's almost like a stickier product associated with that. Both types of products are going to be needed within the body, and we'll find it in different locations. Now, the cells that are involved with serous secretion, secretion of serous products, again, are going to be producing that watery proteinaceous secretion. And so if they're going to be making a lot of proteins, we would expect them to have the characteristics of a protein synthesizing and secreting cells. So these cells are going to have a basophilic staining appearance because they're going to have very active nuclei. They're going to have lots and lots and lots of rough endoplasmic reticulum. In general, they're going to have their nuclei kind of towards the basal third of the cell. They're going to have a lot of rough endoplasmic reticulum towards the middle third of the cell, and they're going to have the secretory product within secretory vesicles at that apical third of the cell. And so because of this, these cells are often described as having basal basophilia. Basal, towards the base of the cell, the foundation of the cell in essence. Basophilia because of the basophilic staining, because of the nuclei and the rough endoplasmic reticulum. You can find these serous secreting cells in areas like the pancreas and the parotid salivary gland, as well as other locations within the body. Again, that characteristic called basal basophilia is a characteristic that's very common for protein synthesizing and secreting cells. They're going to have a euchromatic nucleus towards the base of the cell. They're going to have lots and lots of rough endoplasmic reticulum towards the middle third of the cell, and then secretory granules towards the apical portion of the cell, where they're accumulating, waiting for that signal so that they can be released normally through the process of exocytosis. We take a look at the mucus cells, and again, they're involved with producing sulfated and silated mucins. They're going to have a much paler staining appearance. The serous cells were very basophilic. The mucus cells are going to be very pale in hematoxylin and eosin. We use that periodic acid shift, the PAS stain that we looked at in one of the early slides. They may have kind of a purplish or magenta staining appearance. When we take a look at these, the cytoplasm is going to look relatively pale, kind of purple or pinkish in appearance, and the nuclei in general, instead of the round nucleus that we would see in a serous secreting cells, these are going to be flattened nuclei towards the base of the cell. And so we can see these mucus secreting cells in areas of the body like the sublingual salivary gland. And then the final secretory cell we're going to look at are going to be the goblet cells. And the goblet cells are an example of a unicellular or a one-cell gland. So it's an isolated cell within an epithelium, which is going to be involved with synthesizing and secreting mucus. And it's going to be referred to as a goblet cell because it's going to look similar to a goblet, a wine goblet. So it's going to be attached to the basement membrane. It's going to have a very thin stalk, and then it's going to become enlarged towards the apex where we're going to be accumulating the secretory product. That enlarged region, like the glass portion of the goblet, is going to be where the mucus is going to be stored prior to secretion. So it's going to have a relatively pale staining appearance in hemotoxin and eosin, and it'll look kind of purplish or magenta with the periodic acid shift reaction. We see lots of goblet cells in areas like the small intestine and some portions of the respiratory epithelia in the upper portions of the respiratory tract. And this finishes up our series of lectures on the epithelium. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thanks, and I'll see you with the next lecture.